So, welcome to this class on Neuroscience of Human Movement. This is part 9 of the discussion on primary motor cortex. So, in today's class we will be talking about lesions and what lesions to the motor system at different levels could cause, what effects could they have on the movement patterns in different species. So, how lesions could compromise fractionated control of muscle activity patterns, or in other words fine activity of muscles, fine activity, fine movements, etc. Model for studying this is the pre and post lesion studies on various animals, rats, cats, monkeys, etc. and what could lesion to the pyramidal tract do, and some of the outcomes of lesions including Babinski reflex, but not limited to that. So, there are other things. So, before we move on, we need to define this word lesion. So, this means an experimental or otherwise caused damage to the tissue in general. But in this case we are talking about lesion to a brain part or a region within the brain. If you are saying a region within the brain is lesioned, what we are meaning is that that part of the brain, its structural integrity has been compromised, which also means that its functional integrity or its ability to perform specific functions may be compromised. So, obviously there is structure function relationship. So within the particular area, if there is a lesion, that means there is going to be loss of tissue or tissue damage. This could occur due to pathology that is progressive due to various reasons, or this could be caused experimentally in animals, for example. So, this opens up an interesting approach to study of functions. So, or the structure function relationship. If the structure is compromised, how much or to what extent or which function is compromised are the questions that are being asked. Problems with this approach is a controversial approach. The problem is the temptation to localize a function to a particular area of the brain. For example, if say the primary motor cortex, or if within the primary motor cortex a small area is lesioned, or what is called as a focal primary motor cortex lesion, and let us say that the movement of the fingers is compromised, suppose that is the result of this lesion, then immediately it is tempting for us to make the claim that, oh, you know what, fingers are controlled by that part of the brain, that by that part of the primary motor cortex. What is the problem with this approach? The problem is that it need not be that only that part is responsible or that function is localized within that part. It is possible that this particular compromised region or this lesioned region is forming an important component or a crucial component of a relatively larger network. Or the trivial example that is given is that, suppose you have a TV, and the part that connects to the power, which is basically the plug point, is cut for whatever reason. You are not able to connect to the plug point, then you are not able to connect, and so the TV is not functioning. That does not make the plug point the most important part of a TV. So, it is really tempting to make such claims. One must be very cautious about making claims about localization of function with respect to a compromise of a structure. So, that is a dangerous consequence. Okay. Functional localization is questionable. We do not know if this is true, that is why I put a question mark there, it is questionable. Second problem with this kind of approach, this experimental approach, is that after the lesion is formed, you are measuring movement. So, you perform this kind of studies, which is pre-lesion and post-lesion studies. I have information about movement or measurements, relatively accurate measurements of movements or of a particular movement before the lesion, and then I perform, and then a surgical lesion is inflicted on this animal, and then I perform a study after the lesion, right. Now, once again it is tempting for me to come to the conclusion that any loss of movement function or any loss in say dexterity, in coordination is purely due to lesion. It is very tempting for us to come to that conclusion. What is the problem with this approach? The problem with this approach is that what you are seeing after the lesion is a result of two things. One is lesion itself, the other is adaptation of the organism to the lesion. A great challenge in this field of movement neuroscience is to dissociate these two effects. So, this is the regular question that gets asked in every other seminar. How much of what you are measuring is due to the lesion itself and how much of it is due to adaptation? Have you been able to dissociate? Is a typical philosophical question that gets asked in every seminar. In other words, you do not know what component of the deficit or the movement deficit is caused due to lesion itself. It is important to realize that the organism is still alive and it is adapting to the problem. What you are measuring is the outcome of multiple processes. It is not possible for you to come up with a response with how much deficit is caused only purely due to lesion. It is very tough, it's a very difficult thing to do. So, in general one must be cautious about coming to some of these tempting conclusions. So, we should be cautious about interpreting this data. Having said all this, despite all these concerns and limitations, lesion studies have continued to contribute to our understanding of how primary motor cortex and other motor regions within the brain control movement function. To a great extent, our knowledge has been improved due to lesion studies, but one must still be cautious about making interpretations. So, these are the caveats. Lesions can happen in multiple regions. Where can they happen? In the primary motor cortex, M1, in the premotor areas, that means supplementary motor area proper, pre-supplementary motor area, dorsal premotor cortex, ventral premotor cortex, pre-dorsal premotor cortex, etc., and to the pyramidal tract. Okay. So, an important component of what is the effect of these lesions comes from where the lesion is. So, it can be in M1, which is primary motor cortex, or it can be in premotor area, or it can be in the pyramidal tract. This is called location of the lesion. Now, what else can also happen? The other problem is, are these lesions big or large lesions? They can be relatively large lesions that cause practically an entire motor area to be wiped out, or they can be relatively small or what are called as focal lesions. Focal lesions are small controlled lesions that are delivered to a particular area within a motor area. So, that means, so this refers to lesion location, this refers to lesion volume. Now, another question that naturally arises is, which one of these two is important? Is the location of the lesion important or is the volume of the lesion important? The answer is both are important. So, the response is a relatively complicated function of lesion location and lesion volume, which is the reason we are still continuing to study. So, this is an area of active research. There is not complete conclusion, complete clarity on what causes what, which lesion causes what kind of deficits. Still you could see, for example, cases where relatively small lesions cause relatively complete paralysis, or big lesions sometimes cause very little, very small amount of deficit in comparison. The reason is that's happening because location is important. Where is the lesion happening is important. So, all these things together constitute or give rise to a great amount of debate in this field. So, we still continue to study this topic of lesions and what causes or what lesion causes what kind of an effect. Okay. So, in general, let us talk about focal lesions. For example, focal M1 lesions, right, focal M1 lesions. These lesions usually cause weakness, slowness in movement, loss of precision in movement, loss of coordination, and so on and so forth. So, so it is not going to cause paralysis, but rather weakness and discoordination are more prominent. Focal lesions. What do I mean by that? Focal means small, relatively selective lesions performed in a particular area within M1. So, the rest of the M1 areas remain intact, only a small area within M1 is compromised. In such cases, it leads to selective perturbation, selective disturbance to function of specific muscles, leading to discoordination, weakness, loss of precision, or what I am going to call as imprecise or imprecision, and general slowness. So, this is what happens due to focal lesions. Now, what would happen if the lesion was large M1 lesions? This is usually more devastating and could cause more permanent and more or damages of a higher amplitude. Usually very large lesions lead to complete paralysis of the contralateral side of the body. Of course, even in focal M1 lesions, if the lesion is limited to one side of the brain, then the effects or the deficits are limited to the contralateral part of the body. Obviously, that must have been obvious. Also, so if one area within the motor map, so we discussed the case of motor map or homunculus. If one area within the motor map is compromised, movements that are represented, that are somehow controlled by that part alone are compromised or affected. In particular, the following is a general case, well observed, this, that the distal regions are more affected. So, that means what? That means if there is a focal lesion to the hand and finger areas, what you are going to see is relatively complete abolition of precise, fine, fractionated movements. So, in other words, so what is lost? What is lost is the ability to make relatively independent movements of the fingers and opposition movements with the thumb, or in other words, loss of dexterity. So, when I hold this pen, what you are observing is production of fine forces, finely controlled forces. So, when I write with this pen, one more time, finely coordinated forces are produced on this pen to produce writing. For example, I say writing, there is a particular set of alphabets that are written. I must produce a well timed and coordinated forces. How much skill level you have before the lesion affects how severe the deficit is. So, in general, post the lesion, what is going to happen is the appearance of clumsy claw-like movements, those that you will see in an infant. For example, if you see infants, so I am talking about babies less than 3 months old, for example, or a few weeks old. So, when they are performing flexion movements of the fingers, for example, those movements, what you will see is that all the fingers move simultaneously together like that. Whereas in individuals, in adults, right, you will see individual finger movements relatively independent. Actually, there is less independence within the fingers, so that topic we will reserve for another day. In adults, there is an increased independence of individual fingers that I am able to see. For example, I am moving the ring finger with very little movements of the other fingers. So, this is called as the increased independence. There is a lack of individuation or enslaving that we will discuss. That topic we will discuss in another class. But for today's class, it is sufficient to know that the adults can make more individuated movements. In babies, this is not the case. You can observe in babies less than a few months old that when they are making the flexion movement, and they make flexion movements quite frequently, when they make those flexion movements, all the fingers move simultaneously. So, what is happening is a claw-like movement, relatively clumsy, note that these babies are developing, right. So they are growing and their motor skills are getting developed. As they are getting older, the ability to perform fine actions improves, and the ability to hold a pen and write, for example, appears much later, right, not when they are few weeks old. So what happens when lesions are in the pyramidal tract? So these lesions to the pyramidal tract cause what are called as pyramidal syndrome. Now what is this? Pyramidal syndrome. Now this refers to a set of symptoms, a whole set of symptoms that appear due to larger lesions to the pyramidal tract, but unfortunately these symptoms are also present during lesions of non-pyramidal areas. So there are other cases also where these appear. So it's an imprecise term. Pyramidal syndrome is an imprecise term or a misnomer. Okay, so this usually refers to those lesions that are relatively larger that cause in most cases contralateral paralysis, and so contralateral paralysis is the usual response. There is way more, many more things. It also causes a period of increased muscle tone that is preceded by a period of decreased muscle tone, or hypertonia preceded by hypotonia, something that we have discussed in one of the previous classes where we discussed the reflux cases, right. First hypotonia appears, which is then followed by hypertonia preceded by hypotonia, okay. And it also causes in many cases spasticity. We define this term. Spasticity refers to the appearance of rigidity that depends on the rate at which resistance is applied, or what is called as rate dependent rigidity. So a given load is applied very fast, right. If a given load is applied very fast, then there is great rigidity, and if the same load is applied relatively slowly, then the rigidity is almost 0 or is very limited, appears in very small amount. So this is called as spasticity, and this is distinctly different from general hypotonia. Hypotonia refers to rigidity, increased muscle tone. Spasticity refers to this rate dependent rigidity. One has to be in a position to distinguish the two, okay. What about reflexes? For deep reflexes there is hyperreflexia, and for superficial reflexes there is hyporeflexia, or abolition of superficial reflexes. Sometimes they are completely abolished. And what is also happening is there is an appearance of the Babinski sign. We will see what this is. What is the Babinski sign? We will have to discuss what is the Babinski sign. So in general it turns out that so we said what could happen depending on different species, depending on this. So let us continue the discussion. M1 lesions in humans, M1 lesions, what is the effect if they are focal lesions? If they are focal lesions, then discoordination, loss of fine finger movements and fine motor skills. But in general, as the severity increases, it starts from the problem, the deficit starts from weakness, discoordination, flaccidity, etc., to in the case of large, more specific lesions, in the case of large lesions that are present in a crucial motor area, it could cause complete paralysis. This is in humans. So there are multiple things what we have discussed so far. location of the lesion is important, volume of the lesion is important, but also the species in which the lesion is happening is important. So, this is a topic that we will continue to discuss in future classes. This is also a topic that we have discussed in the past. So, this is a recurring theme. So, the effect that a particular species suffers depends on depends on itself. So, which species it is. So, in other words, if it is a rodent, the effects are less devastating. If it is a cat, it is a bit more devastating. If it is a primate, it is a bit more devastating. If it is a human, it is completely devastating, it is greatly devastating. So, the amount of the severity depends on which species as you go up. So, this. So, why is that? Why is that? This also we have discussed. The answer is, this has and this must have something to do with the presence of fractionated, more individuated control in humans, but not in other animals. Or in other words, we have discussed this earlier, corticomotor neurons or motor neurons from the primary motor cortex make monosynaptic connections to alpha motor neurons that control the fine movements of the distal most parts, or in other words fingers and hand. So, this is more prevalent in humans than in other animals. So, if these neurons or if these corticomotor neurons are compromised, then you must expect loss of fine motor function, right. So, this is the problem. So, this is what happens if the lesions are limited to M1, but we said in the previous slide that it can also happen in premotor areas. So, what will happen if it happens in the premotor areas? Now, that I do not have a slide for this, but I will explain this. So, what is lost in the premotor area? If it is a premotor lesion, what is lost is appropriateness of movement. That makes sense because we have discussed this earlier. The premotor area give context to the movement, whereas the primary motor area gives control, right. So, the best example that is cited in the text and elsewhere is the following. Suppose a candy is placed beyond a transparent glass wall, right, and a monkey is asked to reach and pick up before the lesion. The monkey can realize that what is present is a transparent glass wall, and you will have to go around it. Suppose that is the edge of the wall. Imagine that this is the edge of the wall and there is solid glass behind it, and there is a candy that is placed at this level, right. I am able to see the candy, I would like to pick it up. So, then what the monkey would do is that the hand is here, it would go above the obstruction, pick up the candy and put it in the mouth. This is what a healthy monkey before the lesion would do. If the lesion is to the premotor area, what this would do is that, so there is the, once again the same experiment is repeated after the lesion. So, what happens is that there is the glass wall and there is an edge, and beyond the glass wall there is a candy. Now, the monkey has to reach the candy and pick it up. The monkey will continue to hit the wall instead of trying to go above the wall. I am able to see, so the monkey is able to see the candy, it is trying to reach for the candy. So, in this case what happens is that the natural movement dominates the more contextual movement. The more contextual movement in this particular context is to go above the obstruction and pick up the candy. The natural movement is to think that there is no obstruction and just go straight, right. So, the natural movement dominates the more contextual response. So, the contextuality of the response is lost if the lesion is in the premotor area. Also, that once again more details there is. So, the devil in this sort of work is in the details. What do you mean by that? So, depending on whether the lesion is in the premotor dorsal area or in the premotor ventral area, the response changes. Or in other words, if the lesion is limited to the premotor ventral area or ventral premotor area, right. So, in that case the ability of the monkey to pre-shape the hand depending on object shape, depending on the visual information that I get about the object is lost, is compromised. So, in other words, so responds to visual information about object shape and properties. So, depending on the object's properties, the monkey will pre-shape the hand. That important skill, that important quality is lost if the lesion is in the premotor area or the ventral premotor area. In the dorsal premotor area, contextuality of the movements or the appropriateness of the movements and learned associations are lost. What do I mean by that? Examples of learned associations. So, such as those that refer to learning a change of coordinates, those that refer to learning a visuomotor rotation, for example. Those things are learned associations which will be lost after lesion to the dorsal premotor area. There is more to this picture. So, let us discuss one particular case of the ability of the monkey trying to reach a particular target. The target is presented in presented here in black dashed lines. So, that is the target, right? That is approximately the target area. The monkey has to make wrist movements to reach that area, and they start out. So, that is the starting point, right, or that is the start, and that is approximately the end point. So, in this case, if this is the movement that I want to make, movements are relatively smooth like that. So, these refers to different trajectories. So, these dots here, the green dots here refer to different trajectories, and you see that the movement is relatively smooth and diagonal toward the target. This is pre-lesion, this is before the lesion, right. After the lesion, this is post lesion. After the lesion, the monkey still reaches in some cases, not in all cases, but it is not making diagonal movements, but rather a relatively complicated, more challenging and less optimal, what would be considered less optimal or less than smooth movements are made after the lesion. So, what is lost is the ability to make specific coordinated movements is lost. So, that is our fractionated control of muscle activity has been compromised. So, this also was studied in by Peter Strick's group here, where they talk about where they discuss what happens before the lesion and what happens after the lesion, right. So, this is a few months after lesion, right. So, what you see here, these are different muscles, these refers to flexor carpi radialis, etc., different muscles, right. What you see here, so this is the displacement, this is the movement, right, that is the moment of interest. First, let us talk about the movement. Note the movement is having more definite start and end points before the lesion. After the lesion, it is not clear what the start and end of this movement is. It is not clear for me, or I am not able to make it more, or at least it is not apparent more easily by at least for visual inspection. But here in this case, I am able to, in the case of pre-lesion, I am able to say where the movement started. Well, that started approximately here and ended approximately there. I am able to see and just say, but in this case it is not, there is definitely a start and end, but it seems like that is less pronounced, and obviously this is faster. Also note the amplitude scales, please note the difference in scale. So, there are multiple things, the amplitude is lost and the smoothness is lost. Also note, when you have this sort of a displacement curve, you are going to have a velocity curve that is like that, but when you have this sort of a displacement curve, your velocity curve is going to have multiple peaks. It may have multiple peaks, one has to do the math, but just by inspection I could say that because of this blip here, there is going to be one more peak within the velocity curve. That means what? That means that there is more jerky, less smooth movements. Why is this happening? Well, these movements are a result of muscle activity, coordinated or non-coordinated muscle activity. You could see that the movement onset is at that point, and there is relatively better coordination, and you see before the start of movement this is the activity, after the start of the movement that is the activity. Likewise, this is the activity before and this is the activity after, and so on and so forth. This is the activity before. Now, post lesion, after the lesion, even before the movement starts there is some activity, that is approximately comparable to the activity after the movement start, and so on and so forth. Lots of confusion and also loss of temporal coordination across muscles. So, when you go from to the top to the bottom, or from the top to the bottom, or from the bottom to the top, you are able to observe that the coordination that is present when you view from here is lost when you view from there, for example. There is that loss. Also in a center out reaching task such as this, or in some reaching task such as when you go like that, there is more smooth performance, that is pre-lesion, right. This is pre-lesion. After the lesion you see targets are missed many times, and you see there is greater variability. So, this is like, so before the lesion you see that the variability is that much, after the lesion you see that is that much, and there is more, right. The target is missed here, the target is missed many number of times here, but you see the target. So, obviously, so what that means is that there is a loss of fractionated control, and there is a loss of the ability to coordinate multiple muscles across time. So, the temporal coordination of muscles is lost, and there is more. We said we discussed this spasticity, and we discussed these things, what happens to deep reflexes, what happens to superficial reflexes. This is abolished. Deep reflexes are heightened, not abolished, increased, there is hyperreflexia, and there is Babinski reflex, which we will discuss later. So, before the lesion, this is lesioning to the pyramidal tract, right. So, or what is called as the pyramidal tract lesion or the pyramidal syndrome. So, the monkey is able to hold with the thumb and the fingers, or perform relatively accurate opposition movements with the thumb. So, what is called as a precision pinch grip, like that, right. So, in this case the thumb is opposing the finger, something like this the monkey is able to do, or what is considered to be a more precise movement, that and a more appropriate movement. Please see how the monkey, if this is the candy, so what is seen in pink or rose here is the candy that the monkey is trying to pick up. Please see how it is picking it up, right. So, there is the thumb here and the fingers are here, it is trying to pick up there, like that, a more appropriate response. Here, after sectioning of the pyramidal tract, claw like movements, clumsiness appear. This causes so like that, it will still grasp the object. So, functionally, so when you look at it functionally, it will still grasp the object, take the candy and put it in its mouth, but the details are different. It will, so there is a difference between grasping this object like this and grasping an object like that, obviously, is it not? So, there will be less control or less fine control when I am grasping the object like that. It is not like we grasp this object like this and write. We rarely do that, or nowadays you do not do that anymore. We usually do grasp the object like this. So, fine control is lost after this, is it not? And what also happens is the so called appearance of the Babinski sign or the Babinski reflex. One can Google this and find out. There are some nice pictures from the internet, or the. So, what happens in babies, for example, in infants, when you rub, suppose this is the foot of a baby, suppose consider my palm is the foot of the baby, and I am having a relatively blunt object, right, like that. So, an object like this, and you rub the foot of the baby like that. I am talking about infants, right. Infants, when you do that, suppose these are the toes, when I do that, there will be extension or fanning of the toes, like you see here, there is a fanning of the toes that is apparent in babies. This is considered normal in babies, absolutely normal in babies, and this will disappear as they grow. This will disappear when in adults, when you do the same experiment. So, you take an adult and in his foot you rub a blunt object like that. This will not be the response will be that. So, there will be flexion instead of fanning in adults, but lesions to the motor areas usually cause the reappearance of the Babinski sign in adults like that. So, an example of how mere clinical examination could give information about what has happened. You do not have to do, we must do imaging to get more precise information, but a mere clinical examination or by just rubbing the foot like that, what you see in an adult, if you see rubbing of the foot causes fanning of the toes. If that happens, then that is appearance of the Babinski sign or the Babinski reflex, which is abnormal in adults. So, important to distinguish the situation between infants and adults. In infants, this is considered normal and this will disappear after some time. In adults, this is considered absolutely abnormal, a serious sign, right. So, this is called as the Babinski reflex. By mere clinical examination, you could get a picture of what probably is the problem. There must be more examination, but this is the first examination, right. This is what the doctors would do first. Well, there are other points that need to be discussed, but for the purpose of this class, we will stop here. What we have seen so far is that there is due to lesions, there is a loss of independent or fractionated muscle activity or the fractionated control of muscle and fraction in particular temporal control of muscle activity, coordination, muscle coordination is compromised. And pre and post lesion studies on monkeys have given us information about what kind of deficits could be seen in due to what kind of lesions, and pyramidal tract lesions cause a set of symptoms that are collectively called as a pyramidal syndrome. Unfortunately, this is a misnomer. These kind of symptoms are also present in other cases, and the appearance of Babinski reflex due to lesions in adults is considered abnormal. So, we will stop here and we will continue our discussion on lesions of the primary and premotor systems in the next class. So, thank you very much.